In this video, I want to show you how to find tissue culture protocols for any plant. What is going on, guys? Welcome back to another video. So a question that I get on every single video is like, what is the sterilization time for this plant, or what is the best tissue culture media for this other plant? And a lot of the times I don't know the answer straight away. However, I can find it online, and that is what I'm going to show you today. Alright, so before I show you how to find protocols for pretty much any plant, now this is my rant for today, because a lot of people will say that all these companies that are working on tissue culture, they don't want to share like their secrets, they don't want to share information on how they do their tissue culture on their plants. And I mean, that's fair. They are a business and they want to make as much money as they can, and they don't want to have more competition. So that's something that is understandable. However, there is not really a secret. I can pretty much go online and find protocols, media for pretty much any plant that I want. I just need to know how to find it. So let me show you how to do that. Alright, so I asked on Facebook on which plants do you want me to find tissue culture protocols, and the first comment was tropical gugera. I hope I'm saying that right, or at least close. Species if you like a good challenge. So let's see how much of a challenge that will be. So now the first thing I do is, whenever I'm trying to find protocols for a plant, for like a species in specific, is first I basically go to the taxonomy table, which is basically, you don't know what the taxonomy table is. Basically, we start with a species, where it's like the very specific, like if I'm talking about a plant, like this is just tropical gugera. However, gugera is actually not a species. Gugera is above that, it's a genus. So you have a species, then you have genus. In the genus you have potentially multiple species of plants. They can be there, like this one says tropical. They can even be tropical, but they can also be more template. They can be a combination of both. So we have species, genus, then we go to family, order, class, phylum, kingdom, and then domain. So whenever I'm trying to find protocols for tissue culture plants, first I try to find the species, like the exact plant I'm actually trying to do the tissue culture for, or I'm getting asked questions about. That's the first thing I try to find. If I don't find information about that specific plant, then I move up to the genus. So in that case, it's like you will have, like I say, is the genus. You have the plant, but it can be different plants, but they are very similar, where the protocols should also work for that plant, or at least get you started, get you very close, and then you can tweak your potentially your sterilization protocols. Which sterilization protocols I don't pay that much of attention, and I'm going to talk more about that here in a second. But whenever you have the media protocols, that's where I really pay attention, because that's going to get me started, where I'm going to start with that media, and then potentially later on I will potentially tweak the media, change a couple of things to see if I can improve it. Alright, so let's go online. I even going to do on my phone, which is actually more difficult. Now the reason I was doing it in my phone, well, there are two reasons. One, I couldn't get the screen recording on my computer. I don't know why. The second is that a lot of the times I'm actually have to do this while I'm on a call with somebody. If you don't know, we actually do consultations. We do like phone consultations or Zoom consultations. And sometimes I just get asked these type of questions, are like what is the best protocol for like sterilization or media, and at the moment I need to find it. So a lot of times I'm actually had to do it on my phone, because I'm not in my computer. So the first thing I'm going to do is tropical gugera tissue culture, and this is used on Google, used on like the main Google. Now when you are on the main Google, you're going to find a lot of good information, but there is also a lot of information that is not as useful. Let me show you what. So the first thing, it didn't find the tropical gugera. It basically found tissue culture plants, but this is from Tropico, which is actually a company. So we don't care about that. So it must include Gugera on the search. So we find that the Gugera is actually a jeweled orchid. So that's very helpful because we can actually find information about your orchid tissue culture fairly easy. I actually have one here in the lab. So now when you are here on the main Google, you find some good information, but it can be a little tricky. There are some information that is not going to be very useful. So let's take a look. All right, so I found this website here, or like this publication directly from Google. So here is the publication. Now the way I go to publication is that I cannot read the abstract. I just give it a quick look of whatever they try to find. So I can see that they use seeds from the species of Orchid Gugera, Macrophylla. They were grown on MS media too. I guess it's probably half media in the darkness. And let's see. The plant is obtained. They obtained about, what was it? A capsule. The average plants obtained was like 280 plants. Again, and MS media. This is kind of the media they use. They have 100% success with the media. So it looks good. You potentially would like to read the entire publication. However, I don't really have that much time because the question is what is the best protocol for sterilization and the media. So let's find that out. So introduction is like more like waffle. I don't really care about that introduction. So I go to the materials and method. So I have the plant material. So looks like they use seeds. This is the place that they got it. And the culture establishment, they use a solid half strain MS media. They use 10 grams of sugar. Everything was transferred to an NMR flask, whatever. pH adjusted to 5.7. So you can see that can you slowly start to taking notes on what is the media. So far we have found that they use half MS media. They use 10 grams of sugar. pH is 5.7, 7 grams of agar. They autoclave the medium for 20 minutes. Let's see what else. Doesn't look like they use any hormones on this. I mean, there are seeds, so it makes sense. Whenever you use seeds, they potentially don't use any hormones. So this is very useful. I mean, it took me just a couple of minutes just to find this. And this is how they sterilize the seeds. So the capsules were sulfur sterilized for 15 minutes using domestic bleach for 20 minutes. They were rinsed 3 times in sterile distilled water and immediately placed it, transferred in aseptic conditions. The plants were kept at around 22 degrees Celsius, that is room temperature, in the darkness for the first 2 months. After the period, they were transferred to 16 hours for the period. Kind of the light, I don't really care that much, but because these are publications, scientific publications, they need to know what kind of light and whatever, all these things that they use. They have to be very accurate, but I don't really care that much about that. 3 to 4 months later, liquid medium with the same compositions. The culture grow. You can the plants were cultured to 200 milliliters of the following media. So half strength medium. Now you can see that they use K43 modified Corson medium containing the following. They use a banana, so 80 grams per liter. What else? They use fertilizer. They use charcoal, 2 grams of charcoal, 2 grams of peptone, and 10 milliliters of coconut milk. again 10 grams per liter of sugar, pH the same, 5.7, then 7 grams of agar, also sterilized once again. And again it just talks about the light on it, and then after that it talks about the acclimatization process. Now this paper right here was being very useful on how to sterilize orchid seeds, which is fairly common to working with orchid seeds is fairly common. When working trying to work with tissue, that's a lot more difficult. Trying to find protocols for plants is a lot more difficult, but it's still possible. Let's see if I can find one. Alright, so I found another publication here about medicinal orchids and the uses, tissue culture and potential alternative for conservation. Now again, you have the abstract, you have the introduction, all this I don't really care, traditional uses for medicine, don't really care that much about all this kind of stuff. What I care about is their methods, whatever they did to introduce these plants to tissue culture. So they have a couple of pictures here. Conservation, like in situ conservation, ex situ conservation, the propagation. Now it has talk about seeds once again, tissue culture, propagation, germination of seeds in tissue culture. So this is another option that you can see, and you can potentially get more information on some other types of media they have used. And again, from like potentially multiple types of media, you can work on. I mean, you can do whatever you want. You can take some parts of the media from like the recipes and make your own. You can potentially make even a better media if you take, oh, this person used this kind of vitamins, or like this other one use different types of, or like another sugar, or like a different amounts of sugar, or they use a different types of hormones. So you can take from ingredients from all these kinds of media, and you potentially make your own and make it even better. So now for orchid culture, now propagation, like here, there we go. Propagation of orchids using different explants. So it talks about the potential use for some other explants. Let's see. Use orchids using different explants. In vitro propagation using seed culture is less desirable in many cases, especially for horticultural uses. Now that's why they want to use the clone from the mother plant. So how do they do that? Let's take a look. So here they have some pictures. Okay, so good. During the last 50 years, tissue culture techniques using different explants have been extensively exploited. Large scale. Okay, there we go. In vitro culture in various parts, including shoot tips, flower, stalk, nodes, buds, stems, root tips, and rhizome segments. Now, on this paper doesn't show you exactly how to do it. However, it's giving you a reference. So we have the reference here. We have big or beach, or I don't know how to say the name, 1993. So what do? Is you go all the way to the bottom, to the references, and then try to find that publication. So we have big or beach, I don't know how to say the name, 1993, regeneration response to orchid roots, a study in vitro. So what I'm going to do, I just want to essentially copy and paste this back on Google, look it up, and let's see what we find now. Alright, here we go. So I found the paper. So again, I have the abstract, introduction. Now because this paper is the one that I probably care the most, because it potentially had the information that I need, you probably want to read the entire paper. Probably you can potentially skip the abstract and the introduction, but you potentially want to read everything after that. So in vitro propagation of orchids, right here I don't see much. I can just very quickly look and see. The way that I really quickly look to papers is like, especially if I'm looking for media or stuff like that, I can just see if I can find like any plant growth regulators in there, or if it says anything about MS, or if it says anything about agar, or any media ingredient that I can use as I'm quickly scrolling to the publication. If I can find it, and then I'm going to stop and see what is going on there. If I don't see anything of that, and then I just going to keep scrolling up. All right, here we go. So we have plant regeneration of orchids. Various modes of orchids micropropagations as described as follow. So we have tube tip culture. They are very effective, especially for removing viruses. Now, it's definitely not going to show you how to do the tube tip culture sterilization. That's a bit of an advanced technique where you need to use the microscope. You need to get some practice with that. It is something that if you do tissue culture and you have done tube tip culture, you should know how to do this kind of stuff. So let's see. Now also it is a tube tips from both in vitro and mature plants responding from the formation of buds on medium containing 8.8 microliters of BA and 4.1 microliters of NAA. So that show you what kind of plant growth regulators work with these plants. So we know that BA and NAA seems to be a good. NAA and BA was also more effective for multiplying shoot formations from shoot tips than the single effect of NAA, IAA, or kinetic. So now you know that NAA and BA potentially are better than just NAA, IAA, or kinetic. So potentially, instead of like you are using kinetic, you potentially want to change or replace with a BA. Now we have a leaf culture. Talk about leaf explants. Alright, so regeneration potential from leaf explants from mature plants. We found that the medium supplemented with 66.6 microliters of BA plus 28.5 microliters of IAA. Now it changed. It's not using NAA. Now it's using IAA. Successful micropropagation of leaf explant depends on many factors like nutrient composition of medium, growth hormones, source of the leaves, part of the leaf taking, and most importantly the age of the leaf. Now it also talks about inflorescence stalk or floral buds. Now you can see with some of these plants they have a success in medium containing 1 milligram per liter of BA, 1 milligram per liter of IAA, and 200 milligrams per liter of cecine hydrosate. So you can see that all of these plants, they potentially, they are basically very similar. They're all the same genus, but the media can vary quite a lot. So trying to find a straight answer of what is the best medium for this plant, it can vary a lot. Now, something that I have here, and you can notice, is that it doesn't tell you exactly what is the sterilization protocol. Like, it doesn't tell you that use 10% bleach for like 20 minutes. It doesn't tell you this. It doesn't tell you that. However, I don't even pay attention to that, because even though they may have the sterilization protocol, and then I will follow I will use it as a reference. However, whatever they did, it may not work for me. What I do is I take it as a reference, use the exact same protocol, and let's see if it works. Great. But if it doesn't, you need to learn to analyze what went wrong, because a lot of the times, and it happens a lot, quite a bit to me, especially when back like last year when I started to do quite a few videos on like how to introduce whatever plant. Like I had the Monstera deliciosa, I have some other philodendrons, on how to introduce them to tissue culture. And there were some people that they left some a little bit of a mean comments where they say, I tried to do the sterilization protocol that you say and it didn't work for me. Then and then they will get upset with me and say like, I'm not giving you the like the exact same protocol, or I'm hiding something that because it didn't work for them. But I mean, if it doesn't work for them, I mean, then you need to learn to and analyze what went wrong and work for yourself, because basically whatever worked for me, it may not work for you. To start with, we are actually not working with the same plant. I may have a Monstera deliciosa and you may have a Monstera deliciosa, which is essentially the same plant, but they are not the same plant, because this is my plant, this is your plant. They are not grown the same way. My house may have different growing conditions than your house. The plant, it will be most likely will be the different age. It may have you may have problems with disease problems. They may have fungus. They may have some pests or whatever, where I need to work with that, and I need to decide what will be the best sterilization time and practice with that, because I may do it, it may not work. Whenever I did the Monstera deliciosa, I did it, and then it got contaminated, and then I had to re-sterilize it. So that's something that you need to learn and analyze, that if the protocol that you find online or from me or from other place, you need to understand that it may not work, but you need to learn how to make it work. So if it got contaminated, then you potentially need to increase the sterilization time. If you kill the explant, then you probably need to decrease the sterilization time. So that's something that it just takes practice. However, a lot of times it's very frustrating, because it is a lot easier just to blame some other person. It's like, well, yeah, it didn't work, so you are probably not telling the whole thing. You are probably just hiding something that probably didn't work for me. Alright, so when I just try to find some information just to get me started, I essentially just go to Google, type the name of the species of the plant, and then just type tissue culture, and that most of the times that just get me started. Now, if I really want to dig into the essentials, like the media and whatever people have done with some of these plants, or like all the research, the literature, then I go to Google Scholar and then type the name of the plant. So I have a Gugera tissue culture, and let's see what I found. So you can see that just in the first page I can find many publications about this plant. So it's not really that much of a challenge on like trying to find protocols on some of these plants, but it really takes time, because you have to read a lot. You have to do a lot of reading, and unfortunately a lot of the times you cannot even get into the publication. So let me try to find a good one. Oh, look at see, from Gugera propagation from callus and leaf segments. So this is potentially a very good publication to take a look at. So we have here, let's see. So this is all we get. Propagation of Gugera, and it has the references, but where is the publication? A lot of the times the hardest part is not finding the publication, is actually getting to see the entire publication. Okay, so I'm on another paper here, and we can see the abstract. We can read the abstract. However, unfortunately, a lot of the times we end up with this. We end up with like a to be able to read the entire article, you have to pay, and you can see $39.95. That's a lot of money just to be able to read one publication. That is a lot of money. We as a scientist, we actually don't get paid, but but if you buy these publications, we as a scientist, we don't actually get paid for it. Um, so a lot of the times it's probably best just to see who the author is, and if the publication was done, let's say within the last, I would say the last 20 years, you still have a good chance to be able to reach the authors and ask them to see if they can send you a copy of it. And a lot of the times they will be more than happy to do that for you, because, like I say, they probably they don't make money from all of these, from their publications, like from these websites. Now, there is a way that you can actually get into these publications, and it is if you can, let's see. So right here, this preview is a subscription content access via your institution. So luckily for me, I have access because I still work for my university, so I can type my university information. So I need to find the university. Okay, so University of Tennessee. Now it's going to ask me to log in. I won't be able to log in because I'm on my phone, and then it's going to ask me for like dual security thing that I don't have it enabled on this phone, so I won't be able to log in and read the entire publication. But that's something that you can do, is that if you have a university email, a valid university email, you can log in to your institution and then be able to read the publication for free. Now, if you don't have a university account, a university email, you can get in contact with somebody who has a university account. Maybe you have a college student, somebody that is in college, and then they can potentially use their email to be able to, and they can use their email to be able to give you access. Now, don't be that person and then just tell them, find me this publication, and just like that. Or say that I'm working at trying to find protocols for, let's say, for orchids, and just find those for me. Don't do that. Find the publication yourself, like I did, and then you send them the link, and then just ask them and say please to see if they can get access for you, and then they can then send you the publication that way. But be polite. Don't ask them to do the work for you. You do the work, and then just ask them to see if they can just give you access for the publication. Alright, so now we know that the Kujera is essentially a your orchid. So another way that you can essentially find information is just to go to see information about orchid tissue culture, because orchids are very common. There is a lot of information about orchid tissue culture. Now you also have to be careful when you are on the main, like Google, like I'm going to show you here. So you will find some good information. Like you can see that we have actually, from Plant Cell Technology, we already have a blog on how to do tissue culture with orchids, and I'm going to show you here in a second. So there is a lot of good information, but there are some things that they're just not so useful. So let's take a look. Here. So this is from the Times. This information is just talk about orchids in tissue culture for the general public, essentially. You can see that orchids have only become generally available for purchase in the last few years. Use blah blah blah. Just give you a little bit of thing like a commercial growers that you can essentially use tissue culture to propagate thousands of plants fairly quickly. That's why these commercial growings are using tissue culture for that. However, it doesn't tell you anything about protocols or how they do it. So there is something that is good to give it a read, but it's not going to be very useful. It doesn't give you much information. What I say about all of these, like publications that you may find online, like a newsletters or stuff like that, is that is something that is just good for like the general public to be aware of what tissue culture is, because believe it or not a lot of people still know about tissue culture. You can ask even somebody that works in like I can ask somebody, even a professor that is like in the plant science department, ask them about tissue culture. They don't even know what tissue culture is, and they have a degree, they have a PhD in plant science, and it still doesn't even know what tissue culture is. So this is good. This is more to make people aware about tissue culture and the technology about it. But you can go to like plantcelltechnology.com. This is going to be my blog for today. We have some excellent blogs about tissue culture, how to propagate orchids using tissue culture methods. Now, Angeli or Ryder for the blogs, she does a really great job because she is not only a writer, but she knows about science and she does a lot of research about this. So it gives you a little bit of history about the plants, and then the description of orchids. You can even see it has the classification, all that taxonomy that I was talking about, the kingdom, subkingdom, subdivision, division, class, subclass, order, and the family. So it just gives you the whole entire thing. And then it just tells you about the stems and the roots, the leaves, flowers, parts of the flowers. You can see all the pictures. And then it tells you about the tissue culture of orchids. So it tells you the requirements, what to use, like the tubes, the tissue paper, Erlenmeyer flask, which you can find in plantcelltechnology.com. You can use petri dishes, foils, forceps, scalpel, all of this kind of stuff that you need to actually do the orchid tissue culture, and it gives you a procedure. So use whatever you can do. This is actually tells you about the plant. It's not even just for seeds. It tells you about the plants, and it just tells you what to do. So Angeli has done all the research for you, and now you want to get more information on all of this, because although this is still great, they're still fairly basics. You potentially still want to do even more research, but all of this will essentially get you started. So if you go all the way to the bottom, there are your references and the links, which you can just click on it and read more about it, which is just going to give you more information. But Angeli has done a great job on putting all these blogs together. Now the video is already quite long. However, I still want to talk about this plant because there are a lot of regulations involved. So whenever somebody asks me what is the best protocol, or what is the best media for this plant, there is not really a straight answer, and a lot of people will get upset about it. Then there's like, well, I cannot really tell you about it. It's not that it's a secret. The problem is that there is a lot of regulations with these plants that you need to there is a lot of regulation with these plants with whatever you can use on it. Like a lot of the times, especially you are growing organic, you cannot use many of the plant growth regulators, especially the synthetic plant growth regulators. They are considered as pesticides, so you cannot use them on tissue culture. But a lot of people, they are not aware of that. Now, if this is more for personal use, then you can potentially do whatever you want. You can potentially use whatever plant growth regulators you want. But at the moment you start selling them, and comes all these regulations, you need to follow. Well, you always have to follow the rules, even if it's for personal use. However, you are doing this as a business, you really need to make sure to know all your regulations. Not only because let's say we have the United States, and we have somebody from one state that asks me what is the best media for this plant. Well, it depends on your state, because the media you are going to use is going to depend on whatever state you are. Not sometimes not even only the state, but sometimes even the county you are in. Um, so there are the different regulations depending by county. So the best way to find what media to use on these plants is find whatever you cannot use on these plants. So the easiest way to do it is just by contacting your local extension agent and ask them what is allowed to use on these plants. Don't ask them, hey, what is the best tissue culture media for this plant, because a lot of the times they don't potentially doesn't even know what tissue culture is. Back again, I was saying that a lot of people that are not aware what tissue culture is, even if they are in the agriculture business, they a lot of people don't know. So even these extension agents, they probably don't say well, don't know. Uh, I mean, so you need to tell them or ask them, well, I'm growing this plant. What are the regulations on what I can use on them, with all the pesticides? So we are talking fungicides, herbicides, insecticides, what I can use and what I cannot use on these plants. Also ask them about plant hormones, because they potentially consider them as something separate, although they're by the EPA, I believe, some of them are considered as pesticides. But ask them separate to see if you are allowed them to use them, and which ones you cannot use them. So that's kind of the best answer I can give you whenever you ask me about this plant. It's like, I don't know. It's like I potentially can give you the best way to grow them in tissue culture, but I cannot tell you what are the I cannot tell you that what are the regulations on your state or your county, wherever you're living at. So that's something that you have to do the research. Now, if that is still challenging for you, I mean, we have consultations. We actually have a master class coming up where you can learn how to do tissue culture on these plants, and all the way very advanced techniques where tube tip inductions, the tissue culture with the hormones, and how to remove viruses from these plants, and how to go to all of these potential regulations, so you make sure you are doing all the legal ways. So if you are interested, make sure to check the link down below. So that is potentially the easiest way to go on finding the best information on tissue culture for these plants. So that's something that I really want to address, because I get this all the time, like, what is the best tissue culture media for this plant? Well, I can give you an answer, but it may don't work for you, because you potentially cannot use some of these ingredients on especially the plant growth regulators for this plant. Okay, so sorry I couldn't go to more plants, different plants, trying to find media recipes for all of them, all the ones that I got asked on Facebook. But I mean, a lot of them, they were quite basics, like Syngonium, Bucepulantra, Cryptocron, some of these Anubias aquatic plants, some even carnivorous plants. That's something fairly basics that you should be able to find online. I mean, you just need to know how to do it. Hopefully, with following some of the things that I just told you, where you can just go to Google, find some basic information. But if you really want to dig into it, just go to Google Scholar, find publications about it, and just essentially just read them. I know they are boring. I know they take a long time, but if you really want to find the best tissue culture media, that's something that you have to do it. You have to take potentially several publications, see what some of these researchers have done, and potentially just find the best things from each of them. So potentially you can make your own tissue culture media. So that's pretty much it for today. I hope you have enjoyed it, and I will see you in the next time.
Hey
Goodbye.
everyone, thanks for watching the video. If you're enjoying these tissue culture videos, be sure to follow us on social media for more informative content. And if you're interested in conducting your own tissue culture experiments, make sure to check us out at plantcelltechnology.com for all the products you need to get started. Use the code FP10 for 10% off your first purchase.